Hello, everyone, and welcome to today's session. I am Mala Ganigir with North Dakota State University and Minda Growers Limited, currently serving as chair for the NAPB Graduate Student Association. Thank you all for being here for the first webinar of 2023. We have some exciting webinars lined up for the next few months, so stay tuned. These webinars are also being recorded and will be available on the NAPB YouTube channel. We hope these webinars are useful for you as we continue our journey to enrich your skills in plant breeding. And if you're not a member of NAPB, please go ahead and become a member. So without further delay, I hand it over to John for proper introducing our today's speaker. John.
Thank you, Mala. So today we are very happy to have Dr. Mackill with us. I can personally say that he's probably one of the rock stars in rice breeding. So to formally introduce him, he was a senior director for genetics and breeding of cocoa R&D at Mars Wrigley Confectionery until his retirement last year. He is currently an adjunct professor in the Plant Sciences Department at the University of California, Davis. And he was a principal scientist, rain-fed lowlands program leader at and head of the Plant Breeding, Genetics, and Biotechnology Division at the International Rice Research Institute from 2001 to 2011. He has developed over 20 rice varieties and published research on the genetics of resistance to rice blast disease, submergence, and drought tolerance. He and his colleague identified a gene from traditional rice varieties that conferred tolerance to 2 weeks or more submergence. So this gene, which was designated as Sub1, was bred into several high-yielding varieties. And these resulting submergence-tolerant varieties have been adopted by several million farmers since 2009. In addition to his leadership and research work, he has supervised theses of 24 graduate students. He is a fellow of the American Society of Agronomy, Crop Science Society of America, American Association for Advancement of Sciences, and he is also an honorary fellow of the Crop Science Society of the Philippines. He is a recipient of the Innovation Prize for Agricultural Technology of the American Society of Plant Biologists and the Award of Distinction from the University of California, Davis, College of Agriculture and Environmental Sciences. And he is also a Tech Award laureate for international development. So with that, I give you Dr. Mackill.
Thank you very much, John. And it's really honored to be here, especially with this organization. I think, really happy with NAPB is one of the great professional organizations, and especially talking to webinar for graduate students. So I will start my slideshow. Okay, so I hope you can see that now. There we go. So yeah, as John mentioned, my main association has been with IRRI, the International Rice Research Institute, which is based in the Philippines, and also UC Davis. I worked for USDA ARS in the 90s. So I started basically in the 80s at IRRI as a rice breeder, worked in the 90s at USDA in Davis, then 2001 to 2011 back at IRRI. Most of the work I'm going to talk about today was done during that 2001 to 2011 period. And then I do have some follow-up. I was still involved a little bit after I moved to Mars in 2011. And then, you know, I did get some updates on some of the recent things that have happened with Sub1. But most of this work was done in the early 2000s. So I just wanted to start by mentioning the different kinds of flooding because they all require a kind of different approach or a different mechanism. You can have flooding during germination stage, and so there are actually some genes that confer tolerance to flooding when you actually have flooding on top of seeds that are sown underwater. There's a stagnant flooding, which is really a long-term kind of flooding that lasts for maybe months, and so that's a little bit different problem. But the one I'm going to focus on, and the one that's really probably the most damaging, is on transient or flash flooding, and this is flooding of up to 2 to 3 weeks of the entire plant. So in the case of rice, of course, it's adapted to anaerobic soils and flooded soils, but if the whole plant is underwater, then it definitely goes under stress. And so this happens in a lot of rice growing areas where the drainage is poor. And so when you have heavy rainfalls, a lot of times you get overflowing rivers and things like that happen. In fact, happened recently here in California. And so this happens quite a bit in the rice growing areas of South Asia and Southeast Asia. There's another kind of rice that's really interesting called deep water rice, and that's something different entirely. This is rice where it's grown in areas that are flooded to very high depths, over a meter, and in fact up to several meters in some cases. And in this situation, there are actually varieties that can elongate and stay above the water. And you can see them harvesting or visiting their plots by boats. And in the lower right corner, you could see that they actually have the ability to elongate, and then once the water goes down, they actually knee up and they can form a crop. Generally, these are very low yielding areas, and I would say this kind of rice has declined a lot over the last couple decades in Asia because farmers have been able to grow a post-flood crop, and that gives much higher yield. And they can do that because they have some new irrigation sources in the dry season. And so I would say this kind of rice has gone down a lot, but you can still find it in some of the areas in India, Bangladesh, and so forth. So when I started working on this, of course, we knew that there were some varieties that had tolerance to submergence, this short-term submergence. And most of these varieties came from India or Sri Lanka. FR13A is the classical one that's always cited and had really strong tolerance, but there are some others as well. And we could say all of these varieties were, they're very tall, photoperiod sensitive, not very high yielding, not responsive to inputs. And in fact, in most cases, their quality is not really suitable for the market. And so there are many undesirable characteristics of these varieties, but at least they were a source for breeders to look at trying to improve submergence tolerance in high-yielding varieties. So one of the questions is, how does a variety tolerate submergence? And it's really interesting. It's not intuitive necessarily, but if you look at rice in general, when it gets flooded, it will actually start elongating rapidly. And that's a kind of escape mechanism so that the canopy can be pushed above the water level. But in the case of the tolerant varieties, they actually have a kind of a dormant response to flooding. And so they actually can stay alive underwater. And then once the water goes down, they recover. And you might not see how this would necessarily be better for submergence, but you can see from these plants that the ones that don't have this gene Sub1A, they elongated rapidly. And so once the flooding went down, you can see they're pretty much dead. They basically use up all their reserves, and they can't really grow for more than a few days under this condition. And so this is what happens. And the ones that have the Sub1A gene, they can recover rapidly after the flooding and then go on to normal growth. And we actually did the mapping of this gene and positional cloning, excuse me, with my colleague Pam Ronald at UC Davis and Kenong Xu. We identified the actual gene Sub1A. I'm not going to talk about this because this work, most of this was done by UC Riverside, Julia Bailey Series and Takeshi Fakao. And it's not really relevant to the breeding part, but just wanted to mention that this gene is understood at the molecular level fairly well. And it basically results in this kind of dormancy in response to flooding, as opposed to the rapid elongation strategy. So now let's switch to the farmers' fields in the areas where flooding is a big problem. And I started looking at this in around 2001, 2002, visiting some of these areas in eastern India and Bangladesh. And in areas where submergence was common, they were mainly growing this variety. This variety is called SWARA. And SWARA was probably grown on at least 5 million hectares of rice in eastern India. And it was developed in India originally, but it spread into Bangladesh and Nepal. It was spreading mainly by farmers. It was not being spread really by governments, but the farmers liked it so much, they were basically growing it as much as they could in any place they could grow it. Why were they growing this variety? It was very susceptible to submergence. So why were they growing this? And they had a list of all the things they liked about SWARA. So when you look at this list, you would see, well, there's a whole bunch of things here that all of a sudden the farmers found this, and they thought this was the best variety. And so this is the only thing they wanted to grow. And I'd say one of the key things is the quality fit with what people wanted. The quality was, it had this characteristic of satiety, so that for a given amount of rice, people found that it was very satisfying, kept them full. It was very good agronomically. It had very high stable yields, and there were many good characteristics of this. And so it really became the gold standard for varieties in eastern India. And by the way, gold is related to the name SWARA, so that actually came from the color of the hull, which is a kind of a gold, darker gold color. Not a key important trait for the farmers, but it was just something that was associated with this variety. So when I look at this list and then I think, okay, how do we replace this variety with a better variety as a breeder? I think, okay, that's going to be difficult, because some of these things we can't even measure them in our breeding populations. They can only be really evaluated once you get something on the farmer's field. And so it made me think that, and I know there were a lot of breeders working on this, and they were coming up with some good varieties. But in the end, the farmers were reluctant to change from this variety because of all these characteristics. So it made me feel like, okay, this is going to be difficult to actually improve on unless we have specific traits that we can add to it that would make it attractive. So that really encouraged us to put these two together. Okay, we want submergence tolerant varieties for these areas, but at the same time, we also want all those other characteristics. So let's use the marker-assisted back-crossing approach to introduce this submergence gene. And at the time, now this seems okay. Yeah, of course, this is pretty obvious. But at the time, this was not practiced by any breeders. Very, very few breeders were thinking about using this marker-assisted back-crossing approach. So we started to use this in around 2003. And we picked SWARA as our first one because it was just so popular in these areas. And so we could actually start working on the different varieties eventually when we got funding. So what we basically did is we used 2 to 3 back crosses using background selection, very large populations, so that we could select against anything that had background markers. And we also selected for recombination between the locus and the flanking markers. I think it was very important to do this because we knew that there was some undesirable traits associated with the locus that seemed to be linkage and not because of the gene itself. And so we used this approach so that we could develop the varieties that were at least, I would say, 98 or 99% recovery of the original variety, just to be sure that we would really not have any penalties in any of those traits that we were interested in. We started testing these, of course, once we got the first seeds. And you can see from the pictures below this the difference in survival of these varieties under this artificial flood that we put on in a big tank at Erie. So it had very dramatic improvement in submergence tolerance. The yield advantage we measured was up to 3 tons per hectare after 7 to 18 days of flooding. There seemed to be no penalty in the yields of the variety. They seemed to be normal, just like the original varieties. And so because we used this marker-assisted backcrossing approach, we were able to convince the national governments, which are national and also state level governments, they were the ones responsible for the process of approving new varieties. Normally, a new variety had to go through rigorous testing for several years, and it had to be superior in yield to the best variety. So with this approach, the governments were convinced that, okay, for this approach, we just need to know that it's the same, as good as the original variety, and it has the new trait. So that allowed us to get these varieties fast-tracked for approval. So on the table on the right, you could see 6 varieties that we started with that were popular in these areas, and every one of those was officially released in this fairly quick time period in these countries that we were interested in working in. So we started sending the seed of SWARA and to India and Bangladesh in 2005. That's when we started developing the variety in 2003 because of the very quick way we could get the seed by 2005 and start sending it for testing and multiplication. And so India and Bangladesh started multiplying them and growing them 2006. This, on the left, this trial was the first large-scale trial in farmers' fields. So up to this point, we weren't really sure exactly how this would behave under farmers' field flooding because there's so many variables in the farmers' field. But basically, what we saw in these trials is that under any kind of submergence for more than 5 days, the Sub1 version of SWARA was always higher yielding than the non-Sub1 version, and this yield difference was generally between 1 and 2 tons per hectare. And the result is really dramatic when you get to more than 2 weeks of flooding. You can see that the farmers that were growing SWARA, they lost their crop basically, and the SWARA Sub1 were still producing around 2 tons. And just in perspective, yeah, 2 tons is not an ideal yield, but 2 tons is enough for the farmer to feed their family and also even have some rice to sell or give to extended family. So it's a huge difference between no crop and 2 tons of rice. The one on the right is the variety Samba Mashuri Sub1. This was a little bit later on, and you could see basically the same trend, about 1 to 2 tons yield advantage. And the nice thing about it was that the yield advantage was seen in the whole range of submergence. So even if you have a moderate submergence, if you went to the field, you might not see a big difference between Sub1 and non-Sub1, but the yields were going to be higher with the Sub1 than the non-Sub1 by up to 1 or 2 tons. So it really was quite a significant advance for varieties in these areas. And under no submergence, basically the same yield. All the other traits were the same. So that really encouraged the farmers to adopt this variety. Just a few pictures just to show you some of the more dramatic results. Not every case you would see such a photogenic trial, but the one on the left was the first year we did large-scale testing in eastern India. And this farmer on the top left, you could see that after he had a severe submergence, he was ready to give up, and the researcher said, just leave it for a while. So on the lower left, you could see what happened. It basically recovered and produced 4 tons per hectare, whereas the other farmers, either they had to try to retransplant their field, which is of course expensive, and they won't get as high yield if they do that. In other cases, they would just abandon it. On the right, you can see another big difference of SWARA and SWARA Sub1 grown side by side. This is an example of a submergence in Bangladesh, which happened at a very early stage. And so the areas that were not growing Sub1, they were able to retransplant their field with new seedlings. So they would probably have to go and buy seedlings somewhere and retransplant that. But you could see that the SWARA Sub1 did very well in this area. And so even in these fields which were retransplanted, they're going to be pushed late into the season, and they're not going to yield as high as their normal crop, even after all that effort. So you could still see that without needing any retransplanting, you get a big advantage with the Sub1. And this is another example from Bangladesh, a variety that was developed. We developed this variety at Erie and sent it, and was released in Bangladesh. It was based on an Indonesian variety called Cherang, which is a little bit early duration. And now this variety is becoming very popular because of good grain quality and early duration. And the areas where Sub1 was grown, there's basically nothing. They just abandoned those areas. Another thing that I think was very useful for the Sub1 varieties is there were areas in these regions where farmers didn't plant anything because of flooding. And so they basically left their fields fallow. And then once the rainy season was over, they could either transplant some rice into those areas, or just grow a rabi crop, a dry season crop, pulses or things like that, and just avoid rice completely. So the pictures you'd see here are areas where they didn't used to plant rice at all during the wet season, but because of the Sub1 varieties, they now felt confident, okay, they can go in and plant rice, and it can survive these floods. So the original work that we did on the development of the varieties was supported by the German government. And that was a fairly modest program. As you can imagine, it's not like super expensive, right, to do this kind of marker-assisted backcrossing and develop these varieties. But then once you get them, how do you get the seeds to the farmers? That's actually very challenging. And so around the time when these varieties were showing the promise, that was also when the Bill and Melinda Gates Foundation were getting interested in funding research projects in the CGE system, and they were quite interested in our work on rain-fed rice. I think from their perspective, obviously makes sense. The technology was developed; they just need to get it out, and they can get impact. And so they were really keen on getting impact. So they funded our project called Strassa, which is this project was focused on South Asia and Africa. And so it dealt not only with submergence, but also with other stresses like salinity and drought. But the Sub1 rice was really kind of like at the front of this, because that was where they thought there was a lot of potential, and the drought and the salinity was coming along a little bit behind that. So the Gates Foundation really forced us to really get interested in how to get the variety to the farmers, which traditionally that wasn't part of Erie's mandate. Mainly they would develop the technology and then they would hand it over to a partner and a national program or something. But with this project, we actually started to coordinate directly with the groups that were working on dissemination and seed multiplication. And so this project started basically in 2007. At the early stage, we started sending seeds and having them multiplied by the research stations in these areas. And then from 2007, this scaled up through this project, Strassa. And so they started to do very large-scale seed production and testing. And what I wanted to point out is that because of this fast-track release, the first release of the variety occurred in 2009, And so we started working on the project in 2003. The first release occurred in 2009, which is pretty, like, it's amazingly fast for getting approval of a government to release a new variety. But the thing I wanted to point out here is through this project, by the time they released it, we had already multiplied a huge amount of seed. And so this allowed us to very rapidly get the seed to the areas which needed those varieties. And so that was, it was a little bit of a gamble because we couldn't do the dissemination without the official release. Varieties can spread without being released officially, but it's very difficult because they won't enter the government programs for subsidies and all those things. And so it was really key to get the buy-in from the national governments that they would officially approve these varieties before the production. And then you can see very rapid scale up in terms of number of farmers and area of adoption and seed production. What I wanted to show with this slide is this strassa project was really a huge effort that involved many agencies. And my colleague from India, Umesh Singh, Dr. U.S. Singh, was really a key person to start coordinating with all these groups that were interested in working in the rural areas in eastern India, Bangladesh, and Nepal. And so you could see the number of groups that they were working with in these different places was really huge. And we had not really done much of this before. And so this project was, a large percent, I think, of the strassa funding ended up going towards this effort of dissemination. But I should mention that all of the governments, like the state government and the national governments, were putting a lot of money into the project. They were actually paying for almost all the seed production was paid from them, the dissemination and so forth, and it went through the national systems. And so this close working between these different partners was really essential for this adoption of Sub1. Another interesting thing is about strassa is it not only included Sub1, but also some of the other stresses like drought and salinity. And so Dr. Singh, N.K. Singh, who works at IRI in Delhi, he coordinated a project that was funded by the Department of Biotechnology in India. And so basically they got a whole bunch of breeders in all these areas to start introducing different QTLs into their adopted varieties. And so through this particular project, they've actually released varieties that are now not only some submergence tolerant, but also have drought resistance, more drought resistance, and also salt tolerance. And so this picture on the right shows you a little bit of the genotypic differences we could see in drought. This is not necessarily the near isogenic lines, but just showing you some of the variability that you could see in drought and then below salinity. And then the QTLs for these traits were also included into this strassa project. Now, one of the things that I think has been a little bit disappointing is that we haven't seen enough effort to actually see what is the impact of this technology. What is actually the impact in the farmers' fields of this technology? We could see, okay, you can get 1 to 2 tons of yield advantage in trials in our experiments, and experiments being either on the research station or even our experiments in farmers' fields. But now that we've had adoption of these varieties on a fairly large scale, what is the impact? And maybe I forgot to mention one thing, but one of the strategies why we picked this strategy is that we had a feeling that farmers would not grow anything just because it was submergence tolerant, because submergence doesn't come every year. It comes every 2 or 3 years, or sometimes even longer than that. And so the farmer's focus is really on other traits. So we wanted to develop varieties that could be used either in the submergence prone area or in the normal area without any difference. So the great thing about SWARA Sub1 is that even if you don't have submergence, it does well, and it does fine. And so that's kind of like the approach we picked with all the stresses, that we really didn't want to have any yield penalty from these tolerances. You could maybe show theoretically that even though there might be a slight yield penalty in the end because of these stresses, it's more beneficial to adopt them. But in practice, this was not common. And so we really wanted to emphasize that these varieties have to have the same yield characteristic or more in some cases than the original variety. This is just citing a few recent publications on the impact, and the impact that you actually see in the farmer's field is not quite as high as what we see in experiments, which is not surprising. But at least you can we've actually had some documentation of impact from these particular studies. You can see, for example, 19% yield increase in flooded situations under adoption. In the second paper that was focused on Bangladesh. And so in northern Bangladesh, they had quite high adoption of the Sub1 rice. And I don't know exactly why the 6% yield is translated into 55% higher profit, but I'm assuming it's something to do with both the fact that that's added yield onto there without any additional cost, as well as the fact that if they didn't adopt, they might have to have more labor because they may have to retransplant after submergence. So anyway, the adoption and impact seems pretty good from these initial studies. That graph there on the right is the study from Dar et al. 2017, and basically they showed the high advantage yield of SWARA Sub1 under different years. The second year, to 2013, they had quite significant flooding, and so you could see the impact there. It's pretty high in terms of the yield advantage. But I think so. These are just a few of the reports that I've seen. There aren't many, and I feel like it probably should have been studied a little bit more just to see, what was the real benefit of this. Of course, that's my perspective because of my involvement, but I think for a lot of the people who are working there, it's like, well, it's adopted, it works, so let's move to something else, So I can understand that. Now, I mentioned about this emphasis in the strassa project on yields and dissemination of the seeds. And there's something interesting that came out as a kind of a side, which we didn't originally envision. But my colleague, Dr. U.S. Singh and Dr. Abdelbagi Ismail at Erie, they worked on this to actually try to improve the variety release system and approval system in these countries. And, when we started this project, every country had their own system and they would not look at any other system. So, for example, West Bengal and Bangladesh are contiguous. But they would have a totally different variety system. So in West Bengal, they could grow Swarna as official variety because it was released for all of India. And so it was like their official variety. They had seed production. It went into the subsidy system. This variety spread in Bangladesh and became popular in Bangladesh as well, but it was never officially recognized because it was released in India and not in Bangladesh. And so if they had wanted to release it, they would have to put it through a whole new system. But, the breeder was in India. So there was a lot of this difficulty of where varieties would move because farmers moved them. But then governments, they didn't have the mechanism to really, officially recognize that. And so Dr. U.S. Singh spent a lot of effort to talk to the policymakers in different governments that, if a variety does really well in northern Bihar, it will do well in the Tarai of Nepal. So why not, have a coordination among the different countries that if a variety is approved and performs well in one country, the other country could also recognize that from their work and then adopt that variety as well. Because all these varieties are they're really they're not developed by private seed companies. They're developed mainly by government agencies, and so they're in the public sector. And so I think it was in all their interests to try to facilitate the distribution of these varieties to the farmers in these areas. So they started working on this, and they were able to get a fairly good buy-in from these different countries. And so now they went through. I'm not going to go through this in detail, but basically it was obvious that varieties were moving anyway between countries. So if a variety was developed and tested in Bangladesh, could that variety also be grown in India, especially the contiguous part of India that borders? And so it was a pretty good case that these varieties should be available across these different countries officially, and not just because farmers happen to lived on the border and then moved the seed across. So this was just basically some of the summary, and they actually involved potato as well, because Dr. U.S. Singh was working for some time with SIP, and so he wanted to include potato as well. So they encouraged the governments, and there was agreement of this. And they had several of these meetings, which again, I won't go into the details, but these were meetings where the actual people attending were, in most cases, secretaries of agriculture, and they were approving these policies changes so that they could adopt varieties across the region. And so with each subsequent meeting, they would expand it. So it started with just India, Nepal, and Bangladesh, I believe, focus on rice. But then gradually they started to expand to other countries and also to other crops. So I think out of a kind of a side effort of this strassa project, this came about, and it's really facilitated more widespread distribution of some of these good varieties. And the program they have there is called Seeds Without Borders. So that's what they use to describe it, and seems like it's been very effective. So for conclusions, I just wanted to give a little bit of reflection about this project, where it sits in terms of rice breeding in the whole, and what we learned from it, about its successes and so forth. And I wanted to start with just to mention that, of course, marker-assisted backcrossing is a very conservative approach, where you're not really trying to develop something completely new, but something that, like a farmer would say, okay, yeah, this is just like my old variety, and I like that variety, so I'm going to adopt it. So it's a conservative approach, and there was some criticism of this approach for two main reasons. One is because these mega varieties that we used were grown on very large area, and so the feeling was, okay, this is just going to encourage that farmers will be growing a few number of varieties on large areas, which means they could be vulnerable to any kind of new disease or so forth. It's leading to more uniformity among varieties. So that was one of the criticisms. And then the second criticism was that the actual yield of the varieties were not increased, the yield potential. Instead of saying this is a better variety than SWARA, it has a higher yield, it'll be better for the regular conditions. This is just saying this is SWARA, but with submergence tolerance. So those were the two, I think, criticisms of this project, the main ones. And they both have some validity, right? Because so SWARA is grown, maybe it's grown on too big of an area, but at the same time, our approach was, okay, we've been trying all kinds of other things. They didn't work very well, and we felt the varieties would benefit by having the stress-tolerant varieties. The farmers would benefit by growing them, even if they're still growing the same varieties. I didn't see, to be honest, I didn't see much problem that occurred because of uniformity of the varieties, a fewer number of varieties. This variety SWARA has been grown for more than 30 years on large scale. And really there hasn't been like a catastrophe from farmers growing it. And I could say that with most of the varieties, there were some varieties of rice that came down with diseases after they were released. And so, you have to replace them, which is pretty normal. But many of these mega varieties were grown for long times without having this problem. And the second thing I would say is that because of the ease of doing the marker-assisted backcrossing, you can actually develop these in many different genetic backgrounds. And so, that also, you have alternatives. So farmers have alternatives, and they could select their variety regardless. And I just also wanted to mention that the Sub1 gene is one example where I think Erie now, I haven't had as much contact with Erie recently, but I was told that they included in one of their genes as something that they want to incorporate into the modern varieties. So, because it doesn't have a yield penalty, you can actually put it into irrigated varieties. And, a lot of these irrigated varieties, they could also have flooding, right? They're optimized for very, very favorable condition, but then they can also have flash flooding problems. So having that gene is a little bit of an insurance against that. And then for the second one, the yield potential is not increased. Yes, okay, I agree. We've been working on yield potential on rice for a few decades. And unfortunately, it's just that there's a plateau in the yield, and it's been very difficult to improve that. A lot of the yield improvement that's come has actually been because some of the older varieties their yields are actually declining, and there's been documentation of this in the literature. So increasing the yield potential is a really difficult problem in a crop like rice, where breeders have been working on rice for 100 years, right? And so it's at a very high level of yield already. It's very difficult to make further advances. Yield plateau, okay, I mentioned this already, but with the yield plateau, we can actually add these new traits. And so at least that gives benefit to the farmers, real benefits to the farmers, even if the yield potential is not increasing. So the third lesson, I would say, the breeding priorities need to take into consideration the opportunities for improvement. So what I mean by this is that, like when we discuss at Erie, what is the priority for breeding, the administrators would always say, oh, well, we have to, yield is the number one priority. We have to increase the yield. And if I said, like I want to work on submergence tolerance, they would say, well, drought is more important, so why don't you work on drought and instead of submergence? But the opportunity was really high in submergence because the trait was very simple, no yield penalty. So I think the point I had here is you really have to take advantage of the low-hanging fruit, right? And do the things which are almost like a sure bet, and just not just avoid them because something else might be more important to the farmers. Those things that are important are probably more difficult to solve. So I think you have to balance these two things when you think about what are your priorities in breeding. And then the last one I wanted to mention, and I already commented on this, is that this approach is very quick to develop new varieties. And like we could say, okay, we can develop a new variety in 2 to 3 years. And it's not expensive, right? We can say, okay, you give us $10,000, we'll develop a new variety in 2 years' time. That development is a very small cost of the breeding process if you think about getting the seed out, because that dissemination is very difficult. And I could talk another hour probably on how that was done, because there were so many things that were part of that project, including satellite imagery, delivering to the villages that were infected and so forth. But that was a very expensive part of this project, and the actual development of the variety, and I'm not including all the genetic stuff, which was the basic stuff, cloning, the physiology and everything, but the actual breeding part of it was relatively cheap to do. But, the other parts are difficult and expensive. Okay, a lot of people to to acknowledge. So I just wanted to put this out as some of the key people, especially Abdul Ismail and Umesh Singh, who were my close colleagues for this project, and funding from Bill and Melinda Gates, German government, BMZ, and some other agencies that funded our work, and our collaborators at University of California as well. So that's all. I'm ready to take any questions, John. If I can see some things popping up on the chat, so I'll turn it over to you.
Yep. Thank you so much, Dr. Mackill, for that really wonderful presentation. So we just have a few questions from the registrants before they entered, although I saw some really great questions too from the group chat, from the chat. So first, since we are talking primarily on breeding, so what are the reoccurring challenges that you have faced as a breeder, and how did you overcome those?
Wow, that's a big question. I think I think I already touched on the fact that evaluating what the farmers really want is not always easy. We can measure the yield in our experiments, and we can measure the grain quality, do the tests and all this. But I think it's only when you get something out to the farmers that you really get the real feedback, right? They would say, "Oh, this is great, but the seeds fall off too easily when we go through the field, and we need something that doesn't thrash as easily as that variety." Or there'd be traits here, so many traits that we didn't necessarily see before. So that would be a big lesson, I think, is really to get feedback from the farmers as early as possible in the stage, not just for the objectives, but also once you have something interesting, get it out there. And the participatory approach is something we use very commonly in breeding. So I think that was a big challenge. If I was going to talk to you, I spent my last 5 years working on cacao. I think the challenges there are even different from what we had on rice. It's expensive to grow trees, and it's a really different animal than working on rice. But there would be probably some unique challenges in all these different crops as well.
All right. So before we move on to the questions from the Q&A, one, I think, very important question here is, what will be the future improvements in rice under stress conditions? So basically, what are the future steps that we have to take for breeding stress-tolerant rice, I think?
Okay, good question. I think there are, if I look at the work that was going on recently, that when I left and so forth, there were still many things that we didn't quite understand. There were many opportunities, and we didn't have the time and resources to do them. But I wished I could have done that. Like, for example, when I mentioned flooding, there are different kinds of flooding, and we had potential sources for all of those floodings, like the stagnant flooding. We were trying to combine the Sub1 with the stagnant flooding and all these different things. So there was definitely a lot of scope for improving the stress tolerance of flooding over what we had. And that was one of our key focuses before I left Erie. And it's the same for all of the stresses that we worked on. There were a lot of opportunities that were still not taken advantage of. Of course, the other side of that is that after you use the easy ones like the Sub1, then it's not as easy to use the other things that have maybe smaller effects. It's a little bit tricky. And then how are you going to use them? Are you going to continue using the marker-assisted backcrossing? At some point, how many genes can you really stack? So maybe the future would be where you could actually engineer the crops with a specific gene, and that way, hopefully you wouldn't avoid this problem of linkage drag and increasing having all these other side effects that happen when you do breeding. So I think identifying the genes is still a big opportunity for stress tolerance that you could use directly. At the moment, that's not easy in rice, but golden rice was just released recently, and so perhaps there'll be some opening for transgenic crops in the near future.
All right, so thank you, Dr. Mackill. I'll turn over it now to Mala for the questions we have in the Q&A.
So we have one question online. Is there any alternative source of submergence tolerance available other than Sub1A?
So I haven't seen anything at the same level. We did look at a few of the other highly tolerant varieties, and it seemed like they all had Sub1A, all the ones that we studied. But I have seen some reports of potential sources that are different from Sub1A. So I don't really know if those sources have been confirmed. To be honest, I'm a little bit. I haven't been as close to this work as I used to be, but just most of the time I was there, Sub1A was really the biggest one, and I didn't see anything at the same level. There are some QTLs which have lower effect and that add to Sub1A. So that's why we could see some of the Sub1 varieties had better tolerance under heavy flooding. Like we had, I remember one year we had a typhoon at Erie that just kind of wiped out a lot of our trial, and some of the Sub1 varieties were killed and some of them survived. And so those differences are from other QTLs, and some of them have been mapped as well. So, there are other sources, but I haven't seen it at the same level as Sub1.
There is another question about clonal propagation. So the current breakthrough in rice breeding is clonal propagation. Do you have any intention of advancing this method to your varieties?
Clonal propagation. So normally for the commercial propagation of rice, it's all by seed and fixed lines by seed. It's just like any other self-pollinated cereal. There are some hybrid rice that's grown, but it's mainly in China. We use clonal propagation when we multiplied the early Sub1 varieties, because with the marker-assisted backcrossing approach, you end up with one plant that has all the genes that you want, right? And so how do you get it? How do you get seed of that in a fast way? So you can actually, in rice, you can tear apart each tiller and it becomes its own plant, and so you can really scale up seed production using this. I guess I'm thinking that's what the meaning of clonal propagation is, but it's not really practical, I think, in the farmer's field. I know there's some areas where they do some of this when they're transplanting, but I don't think it's very practical on a large scale. And now, I would say that the emphasis now is try to move from transplanting to direct seeded rice, because the labor requirement is much less with direct seeded rice. And so a lot of the farmers are already moving in that direction, away from transplanting. There is some reports on using different methods to get a seed propagated hybrid that you wouldn't have to cross, but that's a different story. So yeah.
So there is one question about the funding. How may we get the industry like the Gates Foundation involved sooner, so we get both impact and funding at a larger scale?
Yeah, I wish I had an answer for that. The Gates Foundation is like any other donor. They've kind of carried a lot of the CG, for quite a while, and I hear reports that they're getting tired of that, and they might want to move to something different. So a lot of donors get tired, and that's why we had the serious problem around 2006, where we really had some of our donors were cutting their funding, and And the Gates Foundation was a kind of like, they kind of rescued IRI, I think, from fairly drastic funding cuts. Yeah, I think the funding now, a lot of it has to be from the governments, and they are in most cases they are doing that, but not all of them. I mean, India is putting and China, of course, as well. They're putting a lot of money into the research funding, but unfortunately, for some of the smaller countries, that hasn't happened yet. But I don't have an easy answer for that. You just have to look where you can find it, I guess. Yeah.
The next question is about some major challenges. What are major challenges coordinating with so many stakeholders? That is one question. The second question is, what were the limiting factors and how do you recommend future projects to focus on most?
So that was, yeah, it's a big challenge to coordinate with so many groups. I think that for us, there were several things that made that a bit easier. One is that we had the right person who coordinated that. That was Umesh Singh, who had that kind of knack for really dealing with so many groups at the same time, and could easily find the appropriate people. And he got the people involved in our project that IRI had never worked with before, like in state government level and so forth. And so having somebody, a good person like that, who just focuses on that, I think is really helpful. And the other thing I would say is when we were doing this project, it was one of the first agriculture projects of the Gates Foundation. And I think having that association was, a lot of people were drawn to it because they thought, oh, this, the Gates Foundation is doing this, so we want to be part of it, So there was a lot of interest from the different partners to join because of it was a little bit prestigious. In fact, Bill Gates himself, he came a couple of times to visit the project and was really high on it. And then a third thing I would mention, which was kind of interesting, is that we worked on the cloning of the gene with was as a collaboration between myself and Pam Ronald. And that cloning and understanding of the mechanism that enabled us to get a very high profile publication in Nature. And I think when I look back on it, we didn't necessarily need all of that to develop the varieties, right? But having that was, that created a kind of publicity around the project. So I remember when that paper came out, some of the state governments in India were contacting us and say, we got to work on this, That came from a lot of this publicity. So having that kind of, publicity is helpful for getting the people to cooperate. But, surprisingly, I have to say that just working with these partners was really rewarding and really good. And they were just so many of them were just really motivated to help. And I visited some of the projects on the ground that were some of them were NGOs working with 1 or 2 villages. And it was just inspiring to see these people working at the ground level. And something that, as a breeder, I was just developing varieties, but at a CG system, a lot of times you don't necessarily get to go right into the farmers' villages and see that. And so it was really quite inspiring how enthusiastic they were. Now, limiting factors, I think I covered that mostly. This project, I think, if I had thought about this project, what would happen before it did, it happened sort of like what I wanted. So I think normally that, how many times does that really happen? That there were so many things that happened, like the fact that there's no yield penalty, that really was, I hate to say it, but a lot of it was luck, I think, right? It was, I was told that is one thing that's part of breeding is luck that people don't want to talk about sometimes, but there's always that possibility in it. As we go more towards breeding by design, of course, now we want to say, okay, we know all the genes, we want to know which genes we put together, but it's still going to be a while before you eliminate some of those things that are just by chance you get them, you know.
I think we have a big list of questions here, but I see one hand raised from one of our attendees, Khushi Tiwari. Would you like to ask a question, Khushi? Okay.
Okay, can you hear me now?
Yes.
Okay, thank you very much. Again, thank you, Dr. Mackill, for a very nice review. And I'm originally from Nepal. Of course, rice is very important there. And my question is, recently you may have seen a report on perennial rice coming mainly from China. That was a publication. I think it looks quite interesting. And so would you like to comment on that? I know it's a little different than what we discussed today, but what do you see the prospect of that perennial rice and also the hybrid rice, which is so important in China, so much acreage, but probably not as much in India and other countries. So would you comment on those two areas? Thank you.
Okay, yeah, thanks for the question. Yeah, the paper on perennial rice I've read, and it's, to me, it's really a huge breakthrough in breeding, because I was also involved with our own perennial project around 2001, 2002. And I think at that time it was decided that that was going to take so long, and the results were unclear. And so they kind of, nixed it. But this work in China that resulted in perennial rice is pretty amazing, what they were able to do. And it took a long time, a huge effort to do that, to get these traits from wild species introduced into cultivated rice into a form that's actually agronomically working, So it's a huge achievement, I think. There's still a lot of problems with commercializing that in a larger scale. Like where can you actually grow it? Is the environment suitable? Is there enough water in the off season so that you could keep the rice in the field all year long? Is in areas where there's low temperature, of course, it's probably not going to work. In areas where there's very long dry season, it may not work as well. And then the last question I have is, at Erie, we could see that when we keep rice in the field continuously all year long, we just get a buildup of insect pests and diseases over the time. And introducing a period of no crop was really beneficial to controlling pests and diseases. So with perennial rice, the idea is to keep it growing all year round. And so I suspect they're in some of the areas they're going to have problems with diseases and insects. So there's still, I think, problems to work out with perennial rice, but now we have the varieties, and people can try it and see how it works. The original work I know at Erie was focused more on the uplands, because in the upland rice, which is in sloping areas, they had the problem of sustainability and slash and burn system and so forth. So they thought if they had a perennial upland rice, it could stabilize those. But even then, it would have to be someplace where they have enough water throughout the whole year to grow it. So the second question is on hybrid rice. And I don't know the latest on hybrid rice, but it was hard to get it adopted outside of India. Surprisingly, I think the best success I saw outside of China. Sorry, I meant China, but they did get some adoption in India. That was probably where a lot of the work was done, and they had some private companies. I don't know what the current status is of that, I have to say. One of the places where it was very successful is actually in the US. in the southern U.S., the rice company, Rice Tech, developed some good hybrids, and they're adopted widely. And I think part of that was just the kind of environment they were working with and the kind of varieties that were available. It just turned out that bringing in this hybrid germplasm gave them a really big boost of yields. And so that worked very well for them. Surprising in some ways, I think, considering that the costs, labor costs are higher in the U.S., right? So, but they were able to develop ways to produce the hybrid seed, and it's been pretty successful.
Yeah, thank you.
I know we are like past 11, the time is up, but one more last question, which is of my interest as well. So it is about the purity of the seed. What entity is responsible for ensuring the purity or identity of seed that is shared across borders? Did seed sharing in this way led to heterogeneous seed sources?
Yeah, we had to pay a lot of attention to seed quality and seed purity, but we pretty much used the normal way that we managed that, just to try to avoid any kind of outcrossing or any kind of mixtures and things like that. When we sent seeds, we sent small amounts, and then we try to work with partners who are trained breeders and they know how to produce seed, can keep the high quality and avoid any mixtures. And transplanting rice also helps to avoid like volunteers and things like that you get with the seed production. And then there was a pretty strong focus in the project on working with seed producers. And in the place I'm more familiar is in India. They had also they had a lot of emphasis, I think, in their national program on commercial seed production, so that they could supply farmers. The problem is that farmers usually just save their seed, right? They save their seed, and so after several seasons of doing that, they start getting heterogeneity in their seeds. And the seed producers were pretty good at roguing their fields and keeping their purity. So I think they were trying to get the farmers to buy, at least every few years, buy good quality seed and replenish your supply. And it made a big difference. The farmers that were growing seed that had been saved for many years, that they had lower yields and lower quality. So there was a big benefit of having high quality seed. And I think the goal really was to try to have it look a little bit similar to like what we have in the US, where a lot of the farmers, that buying the seed is part of their expense, right? They just order the seed every year, and they know they can get it from a producer who has good quality seed, and so I think they're trying to move more towards that, to have more commercial, and that's why we worked a lot with private companies in the project who were interested in selling seeds. And I think most of them were pretty good at it from my perspective. Obviously there's no guarantee that they would have really good seed, but they were also interested in their reputation as well and having farmers trust them. So so it was pretty easy for us in our project to deal with the problem of seed purity, and we didn't really have that much problem. And then the varieties themselves are also somewhat distinctive. So like SWARA, you could see it by the shape of the grains, and if anything looked odd, you could remove it. So but one thing that reminds me of is that some of these varieties, like Samba, for example, if you take SWARA, it was easy to distinguish SWARA from SWARA Sub1. The reason is that the Sub1 gene was linked to something that changed the hull color of the variety. So you could see the difference by looking at the seeds. With other varieties, you couldn't see any difference unless it was submerged. And so that was something that I was a bit worried about. How would you guarantee that what you're getting is the Sub1 and not the non-Sub1? So I'm not sure how that worked out, but I haven't heard any problem having any problem from that. It's possible that some farmers thought they were buying Sub1 and they may have gotten the non-Sub1. But the objective of the national program was to replace the original varieties with the new one. So now I think all the seed production of SWARNA has to be SWARNA Sub1. I don't think they even allow the regular SWARNA because it was denotified from the list in India.
I know that we have like big list of questions, but we cannot attend each and every question. Please don't hesitate to contact Dr. Mackill with your questions. Thank you very much for such an excellent presentation and for your time. And thank you everyone for joining us today. And don't forget to follow us on the social media and connect with us and stay tuned with our upcoming events. Thank you, everyone.
Thank you.